Hello, everyone, and welcome to our next session in the PML School. Today's topic will be PD11 from the Gabrielsson and Weiner reference, sigmoidal response models. As usual, I'd like to remind everyone that there is a Q&A panel on the right-hand side of the WebEx, and you can use that panel to ask questions during the session. So for today's topic, we are looking at 6 different sigmoidal response models from the Gabrielsson and Weiner reference. The input data set that we have is taken from Hayes and Brown, the growth of leaves, and we're actually fitting a sigmoidal response model to a different type of data set. So on the y-axis, we have water content of the leaf, and then on the x-axis, we have distance. And the resultant plot takes a sigmoidal shape. So what we will be doing is building some different sigmoidal models in Phoenix and fitting them and learning how to code them and then comparing the results. So the different sigmoidal models that are selected, they start with the logistic model, Gompertz, Weibull, Richards, Morgan, Mercer Flodin, and Hill. So these above sigmoidal models show the flexibility of Phoenix models with respect to parameter names and equations. For convenience and comparison, all of these models will be parameterized by alpha, beta, gamma, and delta. And of course, with the Hill model, these are referred to as E0, Emax, EC50, and N. Okay, in the Phoenix built-in interface, the Hill model is included. That's where we have the type Emax, and then you basically make menu selections such as whether or not there's a baseline, whether it's stimulatory or inhibitory, and whether or not the model is sigmoid. And the parameter statements will update accordingly when these selections are made. To do these other types of models, we will then switch to the textual mode and edit the model equations to basically construct the different types of models. Okay? So a bit of background about the logistic model and equation. The logistic model is the simplest sigmoid model. It has a lower asymptote of 0. And the upper asymptote is the maximum y. This model is symmetric about the inflection point. And this can be used to model processes in which the point of inflection is approximately 1/2 of maximum y. Here is the logistic model equation, parameterized by alpha, which is the curve's maximum y value, beta, the curve's midpoint, and gamma, the steepness of the curve. Okay. So the logistic model translated into PML code, an important consideration here when doing these text models, by default the text models are looking for the x variable to be time. So to basically use a custom x variable, in this case distance, we're going to use the statement covariate x. Okay, so that's our starting point. That allows us to read in a column of data that's not time as the x variable. Next, we have the model equation. So in this case, it's y equals alpha divided by 1 plus e to the beta minus gamma times x. Okay? And since we're using y basically as our fitted function variable, we're going to observe y observed of column x equals y plus the epsilon for y, the residual error, where the initial estimate for y epsilon is 1. When we actually build a Phoenix model, by default, and the model makes both structural parameters as well as fixed effects. For individual modeling, and in this case we're just modeling a single profile, it's not necessary to have the structural parameters. Those only apply in the population space where you also have random effects. And so all we really need for this model are the fixed effects. So these will be the alpha, beta, and gamma for the logistic model, okay, along with initial estimates. Okay. The second model that we will consider is the Gompertz model. And this was originally used to model human mortality rates with an a priori assumption that a person's resistance to death decreases as age increases. Applied examples today range from actuarial science to modeling bacterial growth curves. In the Gompertz model, the lower asymptote is 0 and the upper asymptote is maximum y. And this represents a special case of the generalized logistic function. With the Gompertz model, it's designed to allow for a very steep initial increase and then a more gradual approach towards the upper asymptote. So as we approach the maximum y, it's approached much more gradually by the curve than the left-hand or lower valued asymptote. Here is the Gompertz equation. It's another 3-parameter model. Parameterizes alpha, the curve's maximum value, beta, which is growth rate B, and gamma, which is growth rate C. Okay, and here we have the Gompertz model translated to PML code. The same basic design of the model is the same. The only difference here is the model equation has been modified. Okay. The third model for consideration is the Weibull model. So the cumulative Weibull model, also known as the stretched exponential function, it adds a fourth parameter, delta, that allows the inflection point to be modified. So in the Weibull model, the upper asymptote is maximum y, but the lower asymptote can be non-zero. So it doesn't have to be fixed to 0 as in the logistic and Gompertz models. An applied example is to use it to model particle sizes during dissolution of a solid dosage form. The Weibull model equation is presented here, parameterized by alpha, the upper asymptote or maximum y value, beta, the lower asymptote, gamma, the steepness of the curve, and then delta controls the x value of the inflection point. Okay. Here we have the Weibull model in PML code, where the model equation has been updated accordingly. The fourth model is the Richards model. The Richards model is flexible for modeling asymmetric sigmoidal curves. It uses the delta parameter to modify the inflection point. In this 4-parameter model, the upper asymptote is maximum y. Lower asymptote can be non-zero. An applied example is use in growth rate curves. So in the Richards model, alpha is the upper asymptote, beta is the growth rate, gamma is the inflection point, and then delta controls the x value of the inflection point using this exponentiated term 1 over delta. Okay, here we have the Richards model in PML code. And again, these slides as well as the text models will be available on the forum afterwards. The fifth model is the Mercer Flodin model. This was first used to model biological efficiency, for example, responses to the presence of certain nutrients. This model allows for asymmetrical growth, which basically means that the inflection point is not necessarily half of the maximum. It can occur elsewhere. The upper asymptote is maximum y, lower asymptote can be non-zero. This is also used in growth rates curves. The parameters here are alpha, curve's maximum value, beta, the growth rate, gamma is a second growth rate, and then delta controls the x value of the inflection point. Okay. The Mercer Flodin model in PML code with associated equation. And then the last model we will look at today is the Hill model. So the Hill model was originally used to describe kinetics of binding oxygen to hemoglobin. It's now used extensively to model pharmacodynamics. When it's parameterized in terms of Emax, EC50, E0, and so on, this is the classical sigmoid Emax model. In the sigmoidal form, an exponent gamma can be used to modify the inflection point. And you can also use E0 to allow the y-intercept or lower asymptote to be non-zero. Okay, so here we have the classic Hill equation with lower asymptote, upper asymptote, x value of inflection point, and then the delta, which controls the steepness of the curve. Okay. Here's the Hill model in PML code. And this is the same model parameterized with the PD parameters. Okay. All right. At this point, I would like to switch over to the demonstration portion of the model. So I'm going to switch to the Phoenix application. And I will pause here briefly to verify that everyone can see my screen. Okay. Bernd,
looks good.
Great, thanks, Bernd. So here we have the input data set. Again, this is taken from growth of leaves, so we have distance and water content for one profile. This is an exploratory plot of the data, plotted water content versus distance, and this takes the sigmoidal shape. So to start constructing some sigmoidal models, I'll take the input data set and send it to Phoenix Modeling and then Phoenix Model. Okay, and the first model will be the logistic model. Okay. Since we're doing individual modeling, I don't want to use a population mode. I'm going to uncheck closed form and set the model type to Emax. So basically what I'm doing with these steps is I'm using built-in features of Phoenix models to get a model that's close to what I want. And then I will modify it in text mode. So what I'm going to basically do here is set the Emax model, since this is a built-in feature, and I'll use the sigmoid version of the model to construct my equation. Okay. And the PML code is basically written as I go here. Next, I'm going to switch to edit as textual, and then say yes to the prompt. And in the text mode, under setup, we need to bump down to model. So the first aspect of this is we want to use covariate X statement. So this is going to basically allow us to customize this covariate statement reading in from a column. Okay. And once I've done that, what I need to do is also set the terms to Y. So I want my equation to be Y and not E. And I will have y odds of x equals y plus y epsilon. Okay, so now I'm basically defining the model in terms of x and y as inputs. Okay, so I need to map the distance here and then water content in the main map panel. I'll bump back to the model. And then, as I stated initially, we don't need the structural parameters in this case, so I'm going to go ahead and clear those out to simplify the model text, and remove the TV, which stands for typical value, and we'll just simply give them names. So I want fixed effect alpha, beta, and gamma. Okay. Next, we'll go ahead and modify the model equation. So we want to say y equals alpha divided by 1 plus ext. So this ext notation is for e to the x. And then we'll define that as beta minus gamma times x. And then close paren. Okay, so now we have the model equation. We've expressed this as x and y. Coming back to the exploratory plot, so at this point I want to enter initial values for these parameters. Okay, so on the exploratory plot, I can just basically estimate a maximum y value. So I'm going to cap this at 25 for my upper asymptote. For the lower asymptote, this is basically the y-intercept. This looks like approximately 1 as an initial estimate. And I also will naively assume that the slope of the linear portion in the middle is approximately 1, just as initial estimates. So when I come back to the model, my alpha, which is the upper estimate, will be 25, and then we'll set the estimates for beta and gamma also to 1. Okay. I see here on the warnings tab, there's a warning tab. So what this tells me is that there's something in the main mappings that I need to fix. So I need to go to, this main mapping panel is now updated with my custom variables. So my y observed is water content, and my x is distance. And now there are no more warnings. So I can go ahead and run the logistic model. Okay, and so I will quickly look at some of the outputs to make sure that I got a fitted curve here. And the model did successfully converge. I have theta estimates and get results. So that's the logistic model. And what I'm basically going to do here is show a workflow of how to construct these different models as we go. So the logistic model has been created, and I'm going to build on this. So I did a fair amount of work to set up the logistic model, but now that I have it, I can make a copy of the logistic model and paste it back into the workflow. Okay, so many of the selections that I've already made are retained in that copy, and I'm now going to rename the second object as my Gompertz model. Okay. So under the Setup tab model, I'm already in text mode. I already have my covariate X statement as well as my fixed effects. And all I need to do now is modify this equation with the Gompertz equation. So this is going to be alpha times exp to the minus exp to the beta minus gamma times x. Okay, in the bottom panel, you may have noticed there was some red text appearing as I was typing. So it dynamically checks the syntax of the model, and those red statements are kind of a clue as to if there's something wrong. But in this case, I've completed the model statement, and there are no warnings. So I will go ahead and run the Gompertz model. Okay, and I will check some selected outputs to make sure that I did get a fit. And so this is the case. Okay, we'll continue to build the additional models. So the next model is the Weibull model. Okay, I'll make a copy of the logistic model, paste it back into the workflow. And so this will be the Weibull model. So in the Weibull model, I'll start by modifying the equation. So for the Weibull model, we want alpha minus beta times e to the minus gamma times x raised to the power delta. So this caret is raised to the power delta. And I'll close my statement here with the parentheses, close paren. And notice in the bottom panel, there's a red message here. It says basically variable delta is undefined. So I've used delta in the equation, but I haven't defined it as a fixed effect. I can easily remedy this by taking one of the fixed effect statements that exists. I'll make a copy of it and move that down one more line and then replace this copied gamma with delta. So at this point, I have now defined delta as a fixed effect and used it in the equation. Okay. So there are no other warnings. So I'll go ahead and run the Weibull model. Okay. And here we have our outputs as well as fitted curve. Okay, we'll move on to the Richards model. Okay, so this next one will be Richards. We'll go to the model text and write out the Richards equation. So alpha divided by 1 plus exp to beta minus gamma times x. And this entire statement raised to 1 over delta. Okay, I don't have any warnings, syntax error warnings. We'll go ahead and run the Richards model. Okay. Okay, verify my outputs. And we'll continue on to Morgan Mercer Flodin. And don't worry, we will go through a comparison. At this stage, we're just basically building the models. Okay. So here we have Morgan Mercer Flodin. Okay, so I have a new equation here. We have beta times gamma plus alpha times x raised to the delta, all divided by gamma plus x raised to the delta. Okay, so we'll go ahead and run Morgan Mercer Flodin. Right. Check my fitted curve. Okay, everything looks okay. And then lastly, we'll set up the Hill model. So take make a copy and paste back into the workflow. This will be our Hill model. And enter the Hill equation parameterized using alpha, beta, gamma, and delta. So we have alpha plus beta times x raised to the delta divided by gamma raised to the delta plus x raised to the delta. Okay. Okay, so model equation is complete. And we'll go ahead and, in this case, the alpha is actually going to be the lower asymptote. Okay, so I've gone ahead and run the model, and I have a fitted curve here. So this was, we saw here that we're able to, Phoenix is very flexible with respect to model equations. You can basically write any equation as your main model function using PML statements. And we've run through. At this point, we'll go ahead and compare some selected outputs. So what we can do is take selected outputs. In individual mode, we'll take some of these selected outputs and construct some comparison plots as well as overall fit. And this can be accomplished using the append worksheet. So you can basically take a worksheet such as theta, send it to a data tool named the append worksheet, and then send the thetas from basically each of these models to the same worksheet. To save a bit of time, I've actually made a template of these data tools so that I can quickly bring them into Phoenix and join them together. So I'll go ahead and use those now. So I'll go ahead and import. So we'll start with the residuals. Okay. So in my append worksheet, I'll go ahead for worksheet 1, bring in the logistic residuals. Worksheet 2, the Gompertz residuals. I've clicked on my Weibull. Okay. Worksheet 3. So I'm simply dragging and dropping the Weibull model in here and selecting the residual sheet. Okay. Worksheet 4, Richards. And worksheet 5, Mercer Flodin. Worksheet 6 will be the Hill model. Okay, so in this case, I'm taking selected columns, the independent variable, which will be the distance, the dependent variable, which is water content, and then IPRED, which is predicted by the model. Okay, so I can go ahead and execute the append residuals. Okay, and then what I want to do is be able to make a comparison plot. So I've created a plot template called the comparison plot. And for my mapping, I will go ahead and read in the append residuals worksheet. We've basically set up a lattice plot here to compare the different modeling results. Okay, so these are the fitted curves for all 6 of the different models that were fit. Okay. Each plot has a title. So the upper left we have the Gompertz, and then we have the Hill moving to the right, logistic, closing out the top row, and then Mercer Flodin, Richards, and Weibull across the bottom rows. So this is a quick way to visualize the output plots for the various models. And we can see that the 3 parameter models, which are Gompertz and logistic, remember that these have an asymptote, a lower asymptote of 0. So both the Gompertz and the logistic, while they do have a nice smooth fit for some portions of the curve, in the initial part they're underfitting the values, since the y-intercept is likely not 0 in this case. But we can also look at the shapes of the other fits visually here. Okay. Next, I'll go ahead and bring in a comparison of the different thetas. We'll go ahead and bring in the append theta. So the thetas are the final fitted parameters here. Okay. So I want to bring in the theta worksheets from each of my model fits. One, Richards, theta. Morgan, Mercer, Flodin, theta. And then Hill, theta. Okay. We'll go ahead and run this, and we'll get the final parameters estimates for alpha, beta, gamma, and delta where applicable for each of these models. So this source column basically has the model name. Parameter column indicates whether it's alpha, beta, gamma, or delta. Then we have the estimate and coefficient of variation. And then I can use this to construct a summary table. So I'll basically import my theta table template and bring in the results of my append theta to construct this. Okay, if you're curious about some of the data tools, how to make these tables and work with append worksheets and so on, these are presented in our introductory Phoenix Win Non-Lin course. But we see here there's a summary table where model by model we get a listing of the parameter estimate, coefficient of variation. Okay, so this is all 6 models. And if we look here at the at some of the results, some of the parameters have very good precision with the coefficient of variation. Typically, it's desirable to have less than 20% on these coefficients of variation. So a lower number is better for confidence in the parameter estimate. Okay. Lastly, to choose between the models, we'll look at the overall diagnostics. So this can be accomplished by appending the overall worksheet. So we'll go ahead and look at the diagnostics for each of these. So this is overall. Overall for Gompertz. Overall for Weibull. For Richards. Morgan, Mercer, Flodin. And then finally for Hill. Okay. We'll go ahead and run this append worksheet to bring together some of the diagnostics. So the diagnostic outputs include the log likelihood as well as -2 log likelihood. So these are estimates of the overall fit, the Akaike and Schwartz criteria. For all of these initial diagnostics, the smaller or more negative number is considered to be a better fit. The nice part about Phoenix is you can actually sort any worksheet. So what I'd like to do is just basically sort by, I'll start with the -2 log likelihood descending. Okay, so we'll click OK. So based on -2 log likelihood source, the worst fit was the Gompertz model. It had a -2 log likelihood of 39.9, and the best was the Weibull with 27.4. Several of the other models fall in between. Okay. And so based on this, this high -2 log likelihood model, we can consider the Gompertz model is probably not the best fit. This is also supported by the fact that it underpredicted the values near the origin due to not having the ability to fit a non-zero lower asymptote. Okay. The logistic model also had that similar graphic issue, fitting the lower portion of the curve, although it did have good diagnostics overall. The Morgan Mercer Flodin model did have better diagnostics than the Gompertz. Although this model did have a very high condition number. So this is another measure of goodness of fit. The condition number, a target value is less than 10 raised to the power of the number of parameters in the model. So this is a very high condition number for Morgan-Mercer Flodin. And having this high condition number is typically an indication that the model is over-parameterized. If we look at Morgan-Mercer Flodin, it has the high condition number. And if we look at the theta correlation, see if we can identify parameters that are highly correlated. Okay, so here. So on this this correlation tab, the gamma and delta parameters are very highly correlated, 0.994. So this was an issue with the Morgan-Mercer Flodin model. So this leaves us with the Hill model. The Hill model had good diagnostics. They were in the middle of the pack, very good condition number. The Richards model, which had good diagnostics. If we look at, based on Akaike, let's try looking at Akaike next, descending. Okay, so the Richards had good good estimate, good diagnostics, as did the Weibull. The Weibull had very good diagnostics. The Weibull actually had the second best AIC, BIC, and then if we go by log likelihood, the Weibull actually had the best, the lowest value for log likelihood. So with these three models, let's go back to the theta table and look at some of the estimates. So focusing in on the Richards model, which is the fourth, the second from the end on the right, the Richards model actually didn't have the best parameter precision. So if we're looking at the CV percents for the Richards model, and the beta was 24.81, the delta was 31.76, gamma 17.89. So although it did have good diagnostics, the parameter precision was not so good. The Hill model, for three of the parameters, beta, gamma, and delta, the Hill model did have good parameter precision. The alpha was the one with the least precision, but still at 23.33%. And then lastly, the Weibull model also had very good parameter precision for alpha and beta. It did have a bit of trouble with the gamma, but the delta was also had a very good precision. So overall, based on the diagnostics and the parameter precision, the Weibull is a very solid choice for a model from this grouping. So this concludes the demo portion. I'd like to go back to the slides. Okay, so it seems as though unfortunately my slides are stuck. Can Cheryl, can you maybe toggle the presenter back to me? So maybe make Bernd the presenter and then switch back to me.
Chris, you have the ball. Okay.
Okay, very good. Thank you. Thank you so much. Okay, so in summary, we've fit several empirical sigmoid models to the data. We've shown the flexibility of the Phoenix model with respect to parameter names and model equations. We fit the model to the data, and we showed a way to compare results, both looking at the fitted curve, the overall diagnostics, as well as the theta parameter precision. Okay. Next slide, please. Seems I'm somehow stuck. Here we go. Okay. Okay, so we looked at the comparison plot. observed and predicted water content versus distance. We saw that for the Gompertz model, since it doesn't have a lower asymptote, was underpredicting. This was also an issue for the logistic model to a degree. The other parameters, when we added the fourth parameter for the Hill model, Morgan, Mercer, Flodin, and Richards, we were able to fit this better and also get a nice sigmoid shape for the Weibull model as well as the other 4 parameter models. We did a comparison of the thetas, so these are summarized in the slides, and I talked through these in the demo. We also have some final results for Hill and Weibull. Okay. At this point, I would like to open up for the question and answer session. Bernd,
okay.
Do you have some questions?
Yeah, Chris, we've got quite a number of questions. We might not be able to cover everything, but as usual, we will post the Q&A, all the questions and responses on our forum after the event. So maybe we start with a practical question. Is it really required to run all the models every time to choose among the best fit?
Oh, yeah, so you don't need to do this. This is an exercise of where maybe you're investigating a new problem and you're not sure which model is the best fit to the data. We're not necessarily recommending that you have to do this every time you have a sigmoid function, but if it's not clear which model to select for a new study, then this might be a useful exercise to choose which model you use going forward. For many cases where you have a straightforward concentration response curve, you can just use the built-in Hill model for that purpose. But you might be investigating some other type of growth curve or indirect response where you want to modify an equation that's not available in the Phoenix library, and this gives you a good way to select this. And you might, for example, compare your custom model to the Hill model and see if your custom model is a better fit.
Okay. One question around the overall diagnostics. Should the difference in log likelihood or any other diagnostic be weighted against the number of parameters? Yeah, so that's a great question. So the log likelihood is the base overall function, but what you can use are the Akaike and Schwartz criteria. The Akaike and Schwartz criteria actually add a penalty for more parameters in a model. So these diagnostics slightly favor models with fewer parameters. So since this is built in, Akaike and Schwartz is your best choice for picking the best model regardless of the number of parameters. Yeah, one other question regarding the diagnostics, since we are looking at -2LL, are all these models nested models? Some of them are. Some of them are. So the logistic model is the simplest 3-parameter model, and you could consider the Hill model to be derived from that, adding the lower asymptote. And you can also have different nested versions of basically whether you include the exponent or not. So if we go back a bit, a good example of a nested model would be if we use the Hill model. I believe sorry. Why don't I go back to Phoenix briefly here? So when we make the Hill model, this is the model equation where we have parameter delta. But there's also a non-sigmoid version of the Hill model with no delta, which can basically be viewed as a nested version of this model. So the exponentiated sigmoid model is a nested version of the Hill model.
But yeah, that in this context, you can consider these some of these models derived from the base logistic model.
Okay, I think, Chris, we have already run out of time, so may we just finish with a Q&A session, and maybe you can go forward with the rest of the slides.
Okay, thanks, Bernd. So what I would like to also talk about here, again, each of these models will be made available in the Certara forum as text files. You can import them into your Phoenix model, your Phoenix project, and then go ahead and use these models wherever you see fit. Questions and follow-up comments can be exchanged in the forum at support.certara.com/forums. I'd also like to mention we have available now Certara University with a wide range of on-demand and classroom courses. We have introductory, intermediate, and advanced instruction in Phoenix Wind-on-Lin, as well as population modeling using NLME and the IBIBC toolkit. Fundamentals of pharmacokinetics and pharmacodynamics, that's a more theoretical course. Non-compartmental data analysis focused course. Programming boot camp, for example, with third-party applications such as R, there's an R boot camp, as well as a PML boot camp. We also have partner lectures and webinar series, so please visit our Certara University website for more information. Okay, and lastly, I'd like to announce that our next session, which will occur on April the 13th at the same time, analysis of a tissue growth kill model, where we'll look at a tumor cell kill model after acute dosing. And the presenter here will be Bernd Wendt. Okay, so this concludes our today's session. Thank you very much for your time and all of your questions, and we look forward to extending our conversation through the Certara forum.